Hello students. Today we'll continue with next topic, that is structure of antibodies. Okay, now what you should know regarding the structure of antibody and which are the different types of antibodies that we take, and what is the use or function of each antibody. Now antibody will have chains. Okay, so it has two chains. One is called heavy chain and another is called light chain. In the previous class, I have always been mentioning that our immune system produces molecules which are Y-shaped, and I named them as antibodies. And today, you are studying detailed structure of antibodies. So, always you should remember in immunology, suppose one foreign antigen enters human being. So foreign antigen will be there. Now consider this a bacteria. Right now to this figure is quite big, it is just schematic. So this is the bacteria on the surface of which will be various types of antigens. The molecular antigenic molecules will be there on the surface of the pathogens. Now when this pathogen will gain entry into the body, it has to face the immune system, it has to encounter the immune system. So therefore this pathogen goes inside. So the immune system is not going to keep quiet. It means that once the foreign particle enters, then our immune system starts fighting against that foreign particle. Understand? It means that our immune system will respond to the entry. Immune system will respond. So it gives a response to the entry of pathogen. As the pathogen enters the body, immune system will respond to it by producing many molecules. So when against this particular pathogen, many Y-shaped molecules will be produced by our immune system. These Y-shaped molecules keep circulating in the blood, lymph, in the tissues also. So these antibodies which are produced by our immune system now will surround this foreign particle like this. Okay, it will coat the foreign particle by binding to the antigen that is present on this pathogen or foreign particle. Now once the antibody binds to the antigen of a pathogen, then neutralization occurs. It means antigen-antibody reaction results in neutralization. What does it mean? So if this is a bacteria, the surface of it is going to have antigen, and antibody will go and bind to these antigens. When they bind, this antigen-antibody reaction is called as neutralization reaction. Because of which now this pathogen cannot multiply, it becomes harmless, and it cannot infect, it cannot multiply. So that is the importance of this molecule called antibody. Now structure of this antibody itself I am going to explain today. Now, so each antibody will have chains. It will have chains. It is going to have certain bonds which are very important. So these bonds will maintain the structure also. So these bonds will be situated in the So I'm drawing those covalent bonds that are formed in cysteine or cysteine residues within the amino acid. So these are the bonds that are present in the antibodies. Now, what is this antibody? How many types are there? So let's start with this. Now, first, what is antibody? Antibody has another name, and that is called immunoglobulin. And therefore, they can have an abbreviation, that is Ig. Ig. This is an abbreviation given for immunoglobulin. Now again, what are immunoglobulins? They are nothing but a type of proteins itself. Okay. They are type of proteins. They belong to classification of proteins. That is how this antibody will be there. And these are very key molecules. These are effector molecules in the immune system. So these are very much important. Now, immune system, what you came to know is whenever foreign pathogen enters, antigens will be present on its surface, then immune system will not keep quiet, it's going to respond to it by producing more and more antibodies. And that structure itself, you are studying what is antibodies, another name, immunoglobulin, also called as immunoglobulin or immunoglobulins. Here also you can write immunoglobulins, another name is antibodies. Now, what is its chemical nature? What it is actually made up of? Primarily, an antibody is glycoproteinaceous in nature. It means combination of carbohydrates and proteins. Okay. So here, usually carbohydrate will be attached to this particular constant region in the heavy chain. Okay. Now, if you see overall, the overall shape will be Y-shaped. In this, you have totally how many chains? 1 chain, 2 chain, 3 chain and 4 chain. Totally 4 chains are there. So what these chains are made up of? Peptide chains. Okay. Even though it's a glycoprotein, primary structure of an antibody is a peptide chain. So here it consists of 4 peptide chains. But it consists of 4 peptide chains. For all of you. So now this is one peptide chain. This is another long peptide chain. This is a long peptide chain. This is again a short peptide chain. Definitely, if the chain is long, its molecular weight also is supposed to be long. So this is also, but comparatively, compared to this long chain, it's very short. So its molecular weight will be small. So therefore, they have a name. The one which is long, it's called as the heavy chain, and the one which is short is called as light chain. So therefore, it has 4 polypeptide chain, 2 heavy chains, and 2 light chains. Because of this, what can be named? It can be written as or identified as H2L2, 2 heavy chains and 2 light chains. Now, recollect here, this is a peptide. In the first year in the biomolecules, you have studied that a peptide should have ends, N terminal and C terminal. So, therefore, N terminal beginning and another part of the protein chain will be called as C terminal. So, also you can note here, this will be N terminal, C terminal of light chain, N terminal, C terminal of light chain. Similarly, N terminal and C terminal of heavy chain. It's a peptide chain, you should know that it is going to have the ends. But that coming to is represented as, now who in the immune system, which cells in the immune system exactly produces these antibodies. So the cell that produces antibody in the immune system is called plasma cells. So it means that every cell will not produce antibody. Only one particular cell is able to produce the antibodies, and that cell is called as plasma cell. From where this plasma cell comes? This plasma cell in turn is developed from B lymphocytes. This which I have already mentioned in the previous classes. It means that here B lymphocyte, B cell, whatever I was explaining in the previous classes, is the lymphocyte. B lymphocyte is very round and it has a thin rim of cytoplasm, and this nucleus is very big. So this B cell will differentiate and produce a plasma cell whose structure is slightly different here. It's more elongated and larger compared to its progenitor parental cell. Okay. Progenitor or parental. So now this plasma cell, so B cell got differentiated into plasma cell. And this plasma cell is able to produce release from it lots of antibodies. Okay, now this is the thing you should understand. The antibodies are released by plasma cell, which in turn is formed by B cell. What is B cell? It is a lymphocyte. And what is a lymphocyte? It is a type of a granulocyte in WBC. Again, remember, WBCs are very important for defense system of acts as a defense system of our body. Okay. However, structure of antibody was explained by Rodney Porter, which is not necessary, of course, but still you can keep it in mind. These plasma cells, how much antibodies they release? That is a question. So consider now, a pathogen has entered a person and his body is now producing antibodies. Because the number of antigens can be millions, and if the number of antigens entering into millions means even antibodies should be very high concentration. Then how come a single plasma produces so many? No. So that means there should be many plasma cells, okay, which will produce very high concentration of antibodies. Okay. That is called the titer of an antibody. Okay, concentration of an antibody is also called titer of the antibody. So therefore, depending on the amount of an antigen, there can be an increase in the concentration of an antibody so that it will neutralize the antigen. Once neutralization is there, it becomes harmless, then this all what happens? Antibody concentration again comes down in the body. Okay. So this can release antibody at the rate of 2000 antibodies per second. That means within a single second, this plasma cell is able to release 2000 antibodies per second. Likewise, there are many cells. So within an hour, within a day, within 2 days or so, that is, suppose the peak has started in 7th day, then 7th, 8th, 9th, it continues, and therefore concentration can further increase. So that is the meaning that the antibodies are not just formed. What I am supposed to tell is antibodies will not be produced in thousands and hundreds. They will be produced in millions. Okay, and the number of plasma cell also will be more. From where they are formed? From the original cell called B cell. Okay. So class switching should occur. Okay, clonal selection theory, according to clonal selection theory, and the class switching process, the number of B cell will increase, the number of plasma cell will increase, and therefore the number of antibodies also will increase. And that too, it will continuously keep on releasing unless there is a neutralization reaction. Okay, unless the body gets rid of the pathogen. So this was about the role of plasma cell in production of antibody. Now seeing into some of its labelings, if you see. Now here this part, is the one where antigen binds. It means suppose this is an antigen, antibody will be binding at this particular site. The antibody will bind to the antigen in these two sites. Now what are these sites called? Antigen binding sites. So therefore this one is called antigen binding sites. What is antigen binding site? It is a part of an antibody that will directly be in contact with antigen or that directly binds to an antigen. Okay. So therefore this is called antigen binding site. Now already I told this one long chain. This is called as heavy chain. And this one is called the light chain. It is called as the light chain. And you can see here, I drew like this one. Now actually this dot stands for sulfur, disulfide bond it is, because antibodies are known to have these bonds called as disulfide bond. So here there is one disulfide here, so these are nothing but disulfide bonds. So this is a disulfide bond. And N stands for minor terminal and C stands for carboxy terminal. And here you see B is variable region. Every chain has a region called variable region because that is not going to be the same when compared to other antibodies. Suppose this is the variable region. Here, whatever the composition is there, it won't be the same with that of compared to other antibodies present. Okay, it is not just one antibody, many antibodies will be there. So this is the region which will always be variable. So this part region will be highly variable. It means it differs from one antibody to the next antibody. Whereas this region, wherever it is labeled C here, C, it stands for constant region. Means these regions will be same compared to, almost similar compared to other antibodies. So that is called as constant region. So variable region, constant region, variable region and constant region. So that is how this is the part. So this is called antigen binding site, heavy chain, light chain, disulfide bond. Okay. And this one here, so if the antibody needs to be flexible, it's also called neat region. So this was about the structure of antibodies. Now, taking one example, suppose a given antigen is there and it is modified like this, and a push, this is an antigen that is present on the surface of pathogen. It can take different shapes. Okay. Then, now the antibody that binds to it is in this way. So this is in short, this is the schematic way I am writing here. Now this antigen will go and bind to the antibody like this. Okay. Antigen. This complex is called antigen-antibody complex. When antigen combines with antibody, antigen-antibody reaction occurs, and a complex is formed called as antigen-antibody complex. Whenever an antigen-antibody complex is formed, we say that it's a neutralization reaction. Once the neutralization reaction occurs, then the pathogen becomes harmless. Okay, and it will lose its ability to multiply within the body, and therefore there cannot be disease occurring. Likewise, every day and every moment, as and when pathogen enters our body, then continuously our immune system will be fighting against the foreign antigens. So every pathogen can have its own antigen on the surface. It won't be the same. Antigen present on typhoid bacteria will not be the same like antigen present on Vibrio cholera, or will not be the same like antigen present on trypanosoma, etc., etc. So therefore, antigens characteristics may be specific for a particular pathogen. So this was antigen antibody reaction. Now by seeing into this, one more additional term, even though not given in NCERT, you can note it. So here I told that antibody is going to, this is the portion, and whole antigen is not going to bind. Whole antigen is not going to bind. Only a portion it is going to bind. Yesterday I told that the portion of an antigen that binds to antibody is epitope. And the portion of an antibody that binds to antigen is called paratope. It means when antigen binds to an antibody, there is an interaction between epitope and paratope. Okay, the whole, every part of antibody is not involved in binding. Here also, every part of an antigen is not involved in binding. A particular portion here, this portion. Here, this portion. And it falls in which region? Variable region. Variable region. An antigen binding site will not fall in constant regions. Okay. So this, where do these antibodies will be there? In the blood, in the lymph, in the tissues, they can circulate it, keep circulating. Okay. So this completes the structure of human antibodies. The fundamental nature of antibodies are as a protein. So since its overall structure is Y-shaped, and generally schematically it will be represented as Y-shaped molecule. So it means if a pathogen enters your body, which has many antigens, your body also responds in what way? Producing many antibodies to stop them from causing the disease. So next is types of antibodies. Types of antibodies. One, I'll just mention them so that you can remember that there are types of antibodies. One is IgG, another is IgM. Ig means antibody M, antibody G, immunoglobulin M, immunoglobulin G. Okay, they belong to globulin proteins. Now some of the main features of these antibodies I am going to explain now. Now IgG, where do you find? What is the speciality of this antibody? It is supposed to be, remember, keep all the things in mind that is produced by plasma cells in your body, and when it is produced, whenever a foreign particle enters, to prevent it from multiplying, to prevent it from causing a disease, these antibodies will be produced, and they are formed from plasma cells, which in turn are formed from B cells. Okay. So IgG, it is the only antibody that can cross placenta. See here, Suppose this is the womb, you know that there is a dish-shaped placenta, umbilical cord, and the developing fetus. Okay. And mother's body. So here what happens? Fetus immune system is not still developed. In fact, even after the birth for some time the immune system is not going to develop, so it should get the immunity from mother's body itself. How does it get? Mother's body will produce all the antibodies. She produces all the antibodies. Understand? In that, the only one that is capable of passing the placenta, reaching to the fetus, will be only IgG. So what you should know is the only antibody that has capacity to normally pass. Okay, only antigen that has capacity to pass the placenta and provides immunity for the developing fetus will be IgG. Okay. That means the fetus, before the fetus immune system is developed, the IgG is going to pass and pass to the placenta and provide immunity. Okay. So this and one more feature you should know here is IgG is the most abundant antibody. So among all the antibodies, the one which is present in highest concentration, you can tell, or most abundant antibody will be nothing but IgG. Then comes IgM. This GMAD is based on the chain, as well as it's derived from a Greek word. So gamma, like that, epsilon, delta, alpha, like that, based on the unit chain. It is being named IgM next. Now IgM is the main antibody in primary immune response. It is the main antibody produced in primary immune response. So, what is this primary immune response? What is secondary? That I am going to explain in the coming in this class further. Okay. So that is the main antibody produced, whereas in secondary response, IgG will be the main. But however, I am highlighting the one which are very much important. Now next is IgA. IgA antibody, this will be specially found in the milk that is produced within the initial few days after parturition. So once the delivery process occurs, within few days, a yellowish milk will be produced by the mother. That's called colostrum. And this is the antibody that is supposed to be present in colostrum. And if you want to know which is the antibody that is present in colostrum, IgA. Then which is giving immunity to the newborn will be also IgA. Colostrum is responsible, provides immunity to newborn. So IgA provides immunity to newborn. So here, what happens? After the birth, even still the immune system is in process of development. So it requires help of mother's antibody for its immunity. So that antibodies will be present in colostrum. The next, IgD. It will help in recognition by B cell. So it's responsible for B cell recognition, because in an immune system, one cell should interact with another cell. For recognition, this antibody is used. So B cell recognition. Now consider B cell has to point to directly to an antigen, or B cell should bind to the presented antigen by antigen presenting cells. So all these cell-to-cell interaction for recognition, this antibody is necessary. It is present on the surface of B cells. Then comes IgE. So Ig is as a marker in certain conditions. Let me say allergies. If a person suffers with allergy, his body is supposed to have high titer or high concentration of this antibody called IgE. So it's present in high concentration if a person is suffering with allergy. So normal concentration is different, but whenever its concentration becomes high, it means that the person is probably suffering with some sort of allergy. So these are the different types of antibodies. So this is immunoglobulins or antibodies. They are glycoproteins chemically. Oral means actually glycoproteins part of protein itself. So fundamentally they are protein molecules. Within the protein molecule, what type means it will be a glycoprotein. Now next is, I'll be explaining about primary immune response and secondary immune response. And you should know certain terms like encounter. So this is called, what called encounter needs to be known. So this pathogen is entering inside a person for first time. That means before in his life he has never got exposed to this antigen. He or she never got exposed to this antigen or this pathogen. First time it is entering in the body. Then after entering, what happens? It will interact with our immune system. After entry, first time entry, okay, it will face our immune system, you can tell in a common way. When it encounters, that itself is called encountering the immune system. After entering, it has to face the immune system or it should interact with the immune system molecules. Okay, within that, again, blood, lymph, and tissue. So here, once it faces, we call it as first encounter. First encounter. Okay. So you should understand what is the first time entry. You can tell what is that? First time. Then see this second one. What is second encounter? That means first time is entered. Now the same pathogen, say after 1 year, 2 years, or so, depending on the circumstance, maybe even within 1 month or so, the same pathogen enters the same body for second time. Same pathogen enters the same body for second time. Then what we call it as is not the first encounter, we call it now as second. Okay. Then consider the same pathogen enters the same body, probably the same place to drink the public water, the same time. Okay, he's going to drink the water in the railway stations and all at the same time at a regular interval. What happens? The same time, the same condition exists. First encounter, second encounter, then third encounter, then fourth encounter, the fifth encounter, the sixth and the subsequent means how many times it can enter. Okay, so you should now know what is an encounter mean. Entry. After entry, it is going to interact with our immune system. Now, common sense, I already told. Once the pathogen enters in our body, our immune system will respond to it. That's called immune response. Okay. So based on the response, there are two types. The primary immune response as well as secondary immune response. So based on whether it is a slow response or fast response. Okay, whether it is responding slowly or whether it responds fast. So obviously when first time encounter, the immune system will be very slow. Otherwise, it will be very fast in removing the pathogen. So comes the term primary immune response. Primary immune response, and the second one is secondary immune response. Okay, primary immune response and secondary immune response. Now, one by one, I'm going to tell the difference between the primary immune response and secondary. Where do these response occur? This response occurs in the same individual. It doesn't mean primary will be in one individual or second. Both the responses, but what is the difference between them? Depends on the encounter, whether it is the initial encounter or it is a subsequent encounter. Okay. So when do primary immune response occurs? Okay, it occurs due to first time encounter of an antigen. So remember, antigen is abbreviated as AG, whereas antibody is abbreviated as AB. So occurs due to first time encounter of an antigen. Now, if you are told that a pathogen is have entered this person for the first time, means whatever response will be occurring in his body, whatever the immune system will respond to this entry is called as primary immune response. Now, how about this? The secondary immune response will be from second and the subsequent encounters. So here the same thing, but in second and subsequent entry. Entry here, you can use the word encounter. So everything is the same. What you are trying to occurs due to second and subsequent encounters. Suppose pathogen enters for first time, response inside will be primary. If the pathogen enters for second time or subsequent time, any of the subsequent times, then the response is called as secondary immune response. So how does it respond? It responds by producing protein molecules, or Y-shaped protein molecules called as antibody. Produces Y-shaped molecules called as antibody in response to entry or encounter with the antigen. So this is antibody. So this is the first difference between primary response and secondary response. Now, if antibodies are produced, they will reach a particular peak. For example, if this is just a graphical representation here, So right now there is no antibody concentration, but once the response starts, then the peak will form. How much time does it take for formation of this peak? Okay, that means how much time does it take this increase the concentration of antibody inside the person? Okay. So definitely I told first time if anything happens the response will be very slow, even during learning process also. But for second and subsequent time it is going to be very fast. So therefore comes the second difference between primary response and secondary response. What is that? Primary response is antibody peak can occur in 7 days for primary response. Okay. Antibody peak, antibody means immunoglobulin. Antibody peak occurs in 7 days. Got? That means first time a pathogen has gone inside the body, immune system is trying to know all the antigens. No instance, it will bind to the antigen and respond to this pathogen and utilize this. So for this, it will not take quickly in 1 second or 2 seconds. It is going to take sufficient time. At least how many days? 7 days to finally increase the antibody concentration to a very high. So it means that requires 7 days, which is again a little bit slow itself. Comparatively, it is slow. Okay. So it means that first time entry, the immune system will be slowly reacting, taking about 7 days to increase the peak of the antibody concentration. So what about this? It will take lesser time because already it has recognized that's why we say memory response. Okay, it means first time entry is there, then when the same pathogen enters for second and subsequent time, our immune system remembers easily, we say, with the help of memory B cells and T cells. So it knows which is the pathogen that has entered for first time. Okay, so now it can easily recognize when the same pathogen enters for the second time. So when the same pathogen enters for the second time, so it will easily know how to respond it. So quickly the antibodies will be produced. So the antibody peak can reach in a lesser number of days. That is between 3 to 5 days. Between 3 to 5 days. If the antibody peak occurs in 7 days and for secondary response it will occur very fast within 3 to 5 days, the antibody concentration will be very high. So by seeing to this, you should know which is stronger response will be the secondary response. Primary response is a bit slower. Understand? So quickly when antibody peak will be there, then quickly all the antigens will be neutralized. Okay, so that there is no disease. So this is about. So then person will get immunity or not? Definitely. When more antibodies are there, he will get immunity. It means that these antibodies will neutralize the antigen and he will not get the disease. And resistant to disease is nothing but immunity. Next one. To establish immunity, it will take a longer time. And to establish immunity, it will take shorter time. Longer time given already. For antibody peak to occur 7 days, which is much longer time compared to the initial, the lower limit of 3 days. Whereas secondary immune response takes shorter time for establishing the immunity. So here, longer time to prevent the immunity means to become resistant to the disease. Whereas this takes shorter time, is very quick. Got? So this is longer time and this is shorter time. Next is, which which is the antibody produced in primary response. Rather than telling antibody, I will tell which is the main antibody produced in primary response and which is the antibody produced in secondary response. In the response, the main antibody will be IgM. Good. There has a secondary immune response. The primary immune response is IgM, and the secondary immune response is IgG. Got? So that is what you should know. In a primary immune response is the IgM, whereas in secondary immune response it will be IgG. Then coming to affinity between antigen and antibody. Coming back here, suppose this is an antigen binding to a particular antibody here. The affinity is chemical attraction. How strongly an antigen will bind to an antibody? More strongly an antigen binds to antibody, it is better because neutralization can occur very quickly. So in primary response, since the encounter is for first time, our immune system will be very slow. It means that even antibody attraction towards the antigen will be weak. It won't be very strong. So we say affinity is less here. The next difference comes, affinity is less. Affinity is attraction or how strong they bind. Affinity is less between antigen and antibody. So affinity is less. Okay. So interaction of antigen antibody again involves the basic chemical forces, hydrogen bonds, van der Waals forces, hydrophobic interactions, hydrophilic interactions like that. Various types of interactions are supposed to be there even between an antigen and antibody. The next is, here affinity is very high. Affinity is very high between an antigen and antibody. So definitely you can see the points which are written here shows that the second response is a very strong response. It can eliminate the pathogen in very quick way and efficient way. Okay. So therefore, okay, then whatever antibodies are produced in primary response, they decline soon. It means that their concentration in the blood and lymph will reduce quickly. But the antibodies that are produced in secondary response, they remain in the circulation and tissues for a very long time. So they last for longer time. So comes the next difference. Antibodies concentration, antibody titer declines. On the other hand, here antibody lasts for longer time. Okay, you can hear other telling. Antibody declines. Here antibodies last for longer time. So say if it declines for few days in primary response, but the antibodies produced in secondary response can last for a few months also. It means that even for a longer time, the antibodies keep patrolling in order to protect our body against that pathogen which has encountered for the first time. So this is again another difference between. Now next is, when you see all these points, how do you conclude which is of high intensity or which is low intensity response? Definitely. This primary response is low intensity response. Low intensity response. But this one is definitely high intensity response. High intensity response. So these are the main points that shows the difference between primary immune response and secondary immune response. Okay. And also what you should know is for this one, it is not the T cells that produces antibody, it is the B cell. Then what is the role of T cells? So this completes primary immune response. Now you see, as you all know, B cells forms and differentiates, you can tell differentiates into plasma cells, and plasma cells form antibodies. Also comes what next? T cells. Then T cells actually do not produce antibody, but they can help with the B cells. They can stimulate the B cells to produce antibody. That is the function. They don't themselves directly secrete antibody, but they can help the B cells. Such type of T cells are called TH cells. The word H stands for helper cell. It will help the B cells produce antibody. Now, what is the importance of this TH cell is? It is TH cell is reduced in number in a disease. Okay, in a sexually transmitted disease called acquired immunodeficiency syndrome, that is AIDS. So therefore a person suffering with AIDS will die due to lack of immunity ultimately at the end. That is due to reduction of TH cells, which is a very key molecule in immune response. Okay. Coming to next, a theory is there called as lock and key hypothesis. Is not there, but I am just going to explain the lock and key hypothesis. The theory. One more is induced fit theory. Emil Fisher's lock and key hypothesis and Forsland's induced fit theory. These are all two theories. What is the use of this theory? What does this theory explain is? These two theories explains the interaction between antigen and antibody. How do they interact? Now, according to lock and key hypothesis, the antibody will interact with antigen in a similar way how the key will interact with in the same way like the lock and key. That's why the lock and key hypothesis. Okay, it means that structurally there is complementarity between an antigen and antibody. That itself is explained in lock and key. That means rigidity is there. Suppose this is also applicable in terms of even what? Enzymes. When enzymes are supposed to interact by this lock and key hypothesis. But excuse me, what I want to tell is I don't want to take the induced fit theory because what is ultimately known, because induced fit theory is for enzyme substrate also, but induced fit theory is not applicable for antigen antibody reaction. So I stick to this lock and key hypothesis. So what does it explain lock and key hypothesis? The binding of, suppose if this is the antibody and this is antigen binding site. If this is the structure of antigen binding site, then opposite will be the structure of antigen here. So this will be paratope and this is, excuse me, this will be epitope and this is paratope. So they bind in a similar way like how the lock will bind to the key. That is what is explained here in lock and key hypothesis. The same can be even applied between interaction between an enzyme and substrate. So this was briefly about an immunoglobulin as well. So two important molecules which you should know in the immune system is one is antigen and another is antibody. The foreign is an antigen and the antibody is our own molecules to destroy the antigens or neutralize them. Okay. So different types of antibodies being explained and also how is the nature of antigen is being explained. Their interaction is similar to lock and key. How the lock binds, how the key binds to a lock. Similar way it is going to bind. So that was about interaction between the antigen and antibody. And always remember, the first time a pathogen enters, at least it will take 7 days for our immune system to respond to it and produce very high number of antibodies. But when the same pathogen enters for the second time, our immune system will be very quick and therefore antibodies can be increased within only 3 days. That means it will be ready to fight with the disease within the third day itself so that the pathogen is ultimately eliminated, neutralized. Okay. So this completes today's topic of structure of antibody. So what all things to be concentrated in this class will be when it comes to theoretical point of view, structure of an antibody is useful. And apart from this, you should know that which antibody is common in colostrum, which antibody passes the placenta, what is the heavy chain, light chain. Okay. Then what is the difference between primary response and secondary response? Which is better? Okay. Which can give us immunity in a better way is the secondary response. Okay. So if I say if the pathogen is very dangerous, then how is the primary response? Primary response is not sufficient to kill the pathogen. In that case, vaccination is given. Similar to the response here, vaccination is based on the memory response. So further topics will be continued in the next class.